Yes, exactly.
What's the driver? Is it so strongly spurred?
Yeah, I mean, in the end, we don't know. But as I pointed out, I showed that one figure that showed how strong the genetically differentiated the populations are. I think they are very isolated. It's a species that's unusual in its ecology. It occurs on north-facing limestone cliffs, and they get scattered around on mountains. And the habitat is really quite rare and quite, widely distributed in southeastern Spain. So I think they're very strongly isolated, and so there's probably very little gene flow between populations. The experiment suggests that if there were gene flow, the selfing strategy could and should spread based on the experimental data. It's still puzzling as to why that hasn't happened, because in the other I was going to say that it's puzzling because in the other populations, we occasionally, in the self-incompatible populations, we find some individuals that have leakiness, and I sort of glossed over it, but if you were sort of looking closely, you would have seen that in one of the graphs where we excluded pollinators, some of the self-incompatible plants did actually produce some fruits. Almost none, but there were some. And we do find that there is a sort of a leaky incompatibility in some populations too. You'd think they might spread too, but I think the difference is that in those populations there is still high inbreeding depression. And what seems to happen if you have a population that for whatever reason purges its inbreeding depression, and I didn't mention, but the self-compatible population that we used has no inbreeding depression that we could measure, where the self-incompatible populations have high inbreeding depression. And so the introgression of a genome in which much of the inbreeding depression has been purged might be a lot easier than actually the spread of a self-compatibility conferring allele in the context of a population that is actually carrying a lot of inbreeding depression. So it does raise a number of questions as to why once self-compatibility evolves, let's say in range edge populations, they don't always spread rapidly throughout the species range, because that's one of the things that motivated our simulations. It makes total sense when you think about it that if inbreeding depression is purged at the edge of the species range, you'll get self-compatibility evolving eventually. We were sort of not surprised by that result, although that's the one that I emphasized in the talk. We were more interested to say, okay, once you've got that transition, what happens over time? You've still got possible migration across that contact zone between self-compatibility and self-incompatibility. Could the contact zone actually shift? And actually, I didn't have time to explain all the details, but it can shift in both directions, or in either direction, I should say, depending on the amount of recombination that's going on amongst genes under purifying selection, and specifically how much linkage there is between the genes that are involved in conferring self-compatibility and the genomic background of those loci. If there's a lot of genetic load that's been purged in regions close to those loci, then the species can spread, then the self-compatibility system can spread. If there isn't, it's actually quite possible for the self-incompatible populations to reinvade. And so that too is something that's a little bit more complex than I explained. The recombination rates in the genome actually do help to predict what happens after you've got this transition. So the short answer to your question is we don't know why it's not happening. The long answer is that it's complicated by a number of things that probably have to do with not just the ecology, but also the genetics of recombination and the interaction between the self-compatibility locus and genetically loaded loci with deleterious mutations. So it's a little bit more involved than I presented, but nevertheless the prediction is that these populations should be able to, well, this strategy should be able to spread. Yes.
My question is, I understand that why it didn't work also to make specific individuals in the population. One assumption is perhaps that the male produce pollen, so with few males you can fertilize a lot of hermaphrodites, so that's easy. But is there any other option?
Sorry, so the question, are you focusing, asking about the females in these populations, or?
So why the population develop, because the majority of the population develop only male individuals, why they don't develop also females throughout the population, or does it develop?
Okay, yeah, so that is a good question. What we have is mainly a polymorphism where, well, I think the way to understand the system is that the ancestral state in the genus is dioece with separate sexes, and the males have this very effective way of dispersing their pollen with the peduncles, with the stalk-like inflorescences. The females don't have that, and so probably dioece is strongly stabilized in the genus by selection for sexual specialization in pollen dispersal and pollen receipt, presumably. And most of the species are perennials that have probably quite stable populations. The species that we're looking at is the only annual species complex. So there was a transition from a perennial life history to an annual life history, and within that annual clade of, it's quite a complex of lineages. In some of them, separate sexes have actually been there's been a transition from separate sexes to monoece, and what it's been is it's the females that have evolved pollen production, and so that's what these hermaphrodites are. They're effectively females that have evolved a pollen production. Okay, so that's, I think, the way to view this. Now, if you go to the populations in the Iberian Peninsula and measure the pollen production of these hermaphroditic modified females, it varies enormously. Some populations have quite high male allocation, but others have much lower male allocation. And we did an experiment that I didn't describe here that preceded some of the work that we've done more recently, where we went to those populations and actually just simply took, so that was just sort of the hermaphrodites and males, and we took the males away in much the same way that Guillaume did with the dioecious populations. That was a sort of an easier situation because the hermaphrodites were already producing lots of pollen, and we found that the hermaphrodites, when you take the males away, quite quickly evolved increased male allocation. If you go to populations in Morocco near Fez, there are quite a few populations that are almost dioecious. And in fact, on the map that I showed with the pie diagrams of the frequency of males and hermaphrodites, I didn't mention this, but you might have seen that some of the pie diagrams actually had females in them as well. Exactly. And so we've sampled those, and in the field, those females do exist. They still, I mean, that pie diagram is quite old now, but those populations have females in them in the field, but when you grow those seeds from those populations under benign conditions in a glass house, you never see the females. They're always hermaphroditic. So I didn't mention the importance of phenotypic plasticity in sex allocation, but it's hugely important. These, apart from there being a strong genetic component to the variation that we're seeing, there is actually a very strong, much stronger phenotypically plastic element to sex expression. And so you can take a female from the field, put it under different conditions, and it starts producing pollen, and it's just a normal hermaphrodite. And so those females in the field, yeah, they are female phenotypically, but they're actually just on the extreme end of a sex allocation continuum from hermaphroditism towards towards full femaleness. And so I would say that those populations are sort of sub-dioecious. The male frequencies are very high, up close to 50%, and the hermaphrodites are producing very little pollen, and in the field, many of them don't produce any pollen at all, and so they're basically female. And I think it illustrates more or less the transition from a hermaphrodite male strategy to a female-male fully dioecious strategy. I think that's just one extreme of this continuum. In the experiment that Guillaume did, where we actually took populations like that and took the males away, we now see a quick increase in, or rapid increase in the pollen production of the females. Many people have wondered, have asked me, well, the obvious thing now would be to put the males back into the populations, and would you actually then see a reversal of that over a similarly short period of time? Well, we've done the experiment by putting males back into the populations just to see how good the hermaphrodites, the evolved hermaphrodites, are at competing with the males for siring success. They're good at competing with each other, but can they compete with the males? We don't have the results from that experiment yet. We'll have it in about two weeks' time. But I don't think you would see a rapid evolution back to fully separate sexes, because I think the selection intensity is much lower now. It's very advantageous to produce pollen if there are no males. But if there are males, it's not actually that big a deal just to keep producing a little bit of pollen. You can still be very female. And so I think there's asymmetrical selection intensity on these populations. If you take the males away, male function will evolve quickly. I expect that if we put when we put the males back, it'll stay polymorphic like that. Well, the females will continue producing pollen for a long time. But eventually, they'll stop producing pollen, because actually, as you see, if you're going to produce pollen, you want to do it with a male-like strategy. And if you're not dispersing pollen from the end of these long sort of inflorescence stalks, then it's better not to produce pollen at all. And so I actually think the inflorescence stalk in these males is probably the key reason why dioece is so stable across the genus, because it's sort of driving the populations to separate sexes. So I don't know if that has addressed your question, but yeah. What triggers the evolution in terms of the genetics, or? Well, we don't know yet. What we do know is that the ability to produce pollen is already there in the females, because occasionally they produce male flowers. The females don't have a Y chromosome, they're XX individuals, so the ability to produce pollen doesn't require a Y chromosome. So the other thing we know is that it's not just simply some sort of developmental instability that allows the plants to produce male flowers. There is actually, there's a genetic component to this, and I suspect it's probably being maintained in dioecious populations for the same reasons as hermaphroditism is being maintained in the Iberian Peninsula, but at a much lower frequency. I expect that there are one or more loci somewhere in the genome with a major effect on the ability to produce pollen, and perhaps many loci with small effects. And I have a PhD student at the moment who's following up on this by doing quantitative trait locus analysis of the experimental populations, and also an analysis of basically scanning the genome for regions in which the frequency has been distorted by selection, frequency of alleles has been distorted. So we've basically, by using a pool-sec approach, we've pooled between 800 and 1,000 individuals from these populations, and we're about to, we just got the data a couple of weeks ago, to estimate allele frequencies across the genome, looking for those that have been shifted more than you'd expect by chance. And hopefully that'll give us some idea of the genetic architecture of this transition. But at the moment we have no idea. My guess is that there'll be a couple of loci that have had a strong effect, but that there's probably, it's probably a quantitative trait. I was talking about this particular idea with Lukáš last night. for those of you who are interested in sex chromosome evolution, I think it's sort of relevant to the model that has been advocated for the evolution of sex chromosomes in plants, and that is that if plants evolve separate sexes from hermaphroditism, it sort of stands to reason that if you start with a hermaphrodite, some individuals have to become male sterile and others have to become female sterile. And so the canonical model for the evolution of dioece in plants from hermaphroditism is that you have the fixation of a male sterility mutation and the segregation of a female sterility mutation, giving rise to a type of XY sex chromosome system. And both of these mutations should be linked together on what then ultimately is the sex chromosome. Okay, so there is some evidence that from some dioecious plants now that the actual genetics, the developmental genetics of sterility, male sterility and female sterility does depend on at least two loci, and they're linked on the sex chromosome. It's not that surprising. But I think what has not been really picked up sufficiently is that many species don't evolve. I don't think they evolve via two discrete major sterility mutations, but probably evolve initially by a male sterility mutation that invades the population, giving rise to what we call gynodioece, where you have females that express the male sterility mutation and hermaphrodites that don't. So you've got your females and your hermaphrodites. And then what happens is that gradually over time, the hermaphrodites evolve to produce less and less, fewer and fewer seeds, so less and less of their female function, and they become enhanced in their male function. And that's been found in a number of species where populations vary in their sex allocation from the hermaphroditic point of view. So gynodioecious populations, where in some populations there's a high frequency of females, close to 50%, and the males are producing very little, very few seeds, if any at all. So those are at the one, they're a bit like the Moroccan populations of Mercurialis annua. They're at that extreme, they're close to dioece. And then other populations, the hermaphrodites are producing the males are producing many seeds, so they're still hermaphroditic. And I think that suggests a different model, actually, that you get male sterility invading a population, giving females, and the hermaphrodites then gradually evolve towards becoming more male as a result of sort of a quantitative shift, and maybe many loci are involved with sex-limited gene expression. That's sort of a model that's implicit in much of the literature on gynodioece, but it's not really been discussed much from the point of view of what that implies for sex chromosome, or the evolution of sex determination. And I think that really ought to be looked at, actually. You know, taking gynodioecious species that have variation in sex allocation in female function of males, or of the hermaphrodites, what's the genetic architecture of that? And I suspect what we've got there is a single genetic locus that gives rise to sex determination, and many other quantitative many other loci with quantitative effects on sex allocation. And so that's sort of vaguely relevant to the whole evolution of sex determination in plants and sex chromosomes. So it might be that we've got quite different models of sex chromosome evolution in different systems that have evolved via different routes. So at the moment, I think there are competing hypotheses. We've got very little data to distinguish them, actually. A few examples where we seem to have these sterility mutations that have been characterized in about 2 species now at a functional level. But we're sort of far behind the animal community in pinning down which loci might be involved and what the genetic architecture is. So, yeah. Sorry, a species that has? Yes.
why it is there, so both pathways can be triggered. But not only necessarily be genetic modification, but can be also be genetic. So one is involved in science, then triggers somehow the event specific pathway, or the other copy, or the other copy is silence or not, then triggers the second pathway. We also explain about these different parts of it.
Yeah, so people have suggested that perhaps the results that we got in the selection experiment point to maybe a role in some sort of enhanced epigenetic regulation of pollen production, possibly. I mean, clearly what's going on is, we're seeing something that's evolved very rapidly. Maybe it's evolved through some sort of epigenetic imprinting that's being retained over generations. We don't have any evidence for or against that, but I guess in the short term that sort of mechanism may well be may well be possible. Yeah.
I would like to ask about expression data from transcriptomes, so were they from leaves or
Yes,
flowers?
those were from leaves actually. Yes, from leaves. Yeah. Yeah.
That's correct. Because if they were from flowers, yeah, it would be like evident that they
Of course, yeah.
transcriptions.
Yes.
And then I would like to ask if you about these increased fitness in self-link individuals. So do you think that it can be just in short-term evolution, however in long-term, like the outcrossing populations have still like higher stability? So
Okay.
Otherwise, keeping the outcrossing
Yeah. So
core of the range.
no, that's a question that did trouble us when we were trying, interpreting the results from the empirical selection experiment. We see this rapid transition to almost the fixation of self-compatibility, but what about the evolution of the system when inbreeding depression sort of evolves with the mating system over longer periods of time? We addressed that actually in this study, and I didn't show any of those data just for lack of time, but we've done simulations of this process using the sorts of variable, estimates of the variables from our experiment, because we were interested in what happens over a few more generations, just basically over the next 20 or 30 generations. Would you then see that just with recombination within the genome, you actually then get the re-establishment of self-incompatibility? Maybe it actually spreads back. And then also in the much longer term, over hundreds or thousands of generations, as you get the possibility of the purging of any inbreeding depression from the mixed populations. And in fact, in almost all of the simulations, and we explored quite broad parameter combinations, we don't see a transition back to self-incompatibility. So yeah, we're still, why doesn't it happen more often? And maybe it does happen more often. The critical thing is that initially you need to start with a population in which inbreeding depression has been purged. And perhaps once that's occurred, it does open the floodgates for the wider spread of self-compatibility. And there are plenty of examples where the whole species is actually self-compatible and highly selfing. And presumably those cases, at least in some cases, selfing evolved in part of the species range and then spread. And maybe this is an example of a situation where initially the ecology and the genetics allows the evolution of self-compatibility, self-fertilization, and the purging of inbreeding depression locally, and then after that settles down locally, it's able to spread into situations where that might not have been able to happen initially. So that's the sort of the way we think about it at the moment, that this is maybe an example of how selfing could spread across a species range even where through parts of the species range it wouldn't normally be selected, but if it's kick-started in some part of the species range, perhaps it can then spread. But I'm still a bit skeptical, as you obviously are too, that this, that why doesn't it happen, or why hasn't it happened in this species? Perhaps we're just seeing cases where the process has started and hasn't yet gone to completion, and with enough time, these populations, these selfing populations or strategies will spread. So, yeah.
Is there any clear pattern or clear difference in amount of phenotypic polymorphism, not genotypic polymorphism, but phenotypic polymorphism between self-compatible and self-non-compatible populations? And similar question, is there any difference in evolvability, ability to respond to
Okay, the first question is, are there any phenotypic differences between the self-compatible and self-incompatible populations beyond just the self-incompatibility itself? And indeed, we've looked at that, and there are very subtle differences, but almost none. They are still, the self-compatible plants are still investing heavily in nectar production. Flower sizes are the same. Pollen-ovule ratios are the same.
Sorry, I don't mean qualitative difference, but quantitative difference in amount of phenotypic variability.
Oh, variability.
Yeah, variability.
So how variable are the populations,
Yes.
are the individuals within a population?
Yeah.
I don't think there are any differences that are obvious enough. There is a difference in the amount of genetic variation, and that you'd imagine would be expressed in differences at the phenotypic level, but we've not been able to pick up differences in the variance, for example, between these populations. They're quite variable, and
I would predict that there will be opposite relation. I believe that in this self-compatible population, less genetically polymorphic one, there will be more phenotypic variability than in this self-incompatible population.
because of
Canalization and effect of heterozygosity and so on.
okay, reduced canalization.
Yeah.
Okay, as a sort of a deleterious effect of potentially deleterious effect of reduced. We don't have any evidence for that, for or against it, but yeah.
And what about this evolvability?
Yes, the evolvability. We've actually tested that in the other system that I presented. So if you remember, in the Mercurialis annua case where we've got the transition between sexual systems, there was the range expansion from southern Spain and Morocco into northern Spain on two different colonization fronts, and we found patterns in, there was less genetic diversity in the north, there was more inbreeding depression, so less inbreeding depression, and increased genetic load and so on. So we see all of that. And so what we did as part of that study, and I didn't present the data, but we measured responsiveness to selection, evolvability in 20 populations along the colonization front. And indeed, as you would predict, the southern, and so how did we do that? We took the population, and we were interested in, particularly in the variation in pollen production by the hermaphrodites. That's something that we could easily select artificially. So the way we did that experiment was in 20 populations, we grew up plants, and we then selected in each of those populations the top 20, or was it the top 10% and the bottom 10%? I think it was the bottom 10% and the top 10% of individuals with each of those populations with respect to how much pollen they were producing. So it was sort of like strong artificial selection in each of those populations. And then we measured the response to selection, one generation of strong artificial selection. And we find that there was a pretty strong response to selection in the southern populations and no response to selection in the northern populations. Do you mean that? Clearly, yeah. Of course. So you're suggesting the longer term, do they have... Did you think it would be the opposite relation, that the northern populations will evolve? So why? Because initially the northern populations will have lower genetic variation, and so presumably lower ability to respond to selection.
There is less episodic interactions. There will be higher amount of
Additive genetic variation. Well, we used our experiment, in a very crude way to estimate, just with that one simple generation of selection, the additive genetic variation. And clearly, as it was much higher in the southern populations than the northern ones, the additive genetic variation, simply using the breeder's equation by inferring how much additive genetic variation there was. But in the longer term, I would predict that gradually you would get you would get increase in genetic variation through migration and perhaps slowly by mutations as well, and the populations would be able to evolve. I think that's but I'm not so sure whether the difference in evolvability is going to be the result of differences in the importance of gene by gene interactions, which I don't know. I mean, is there good evidence for that from increased evolvability as a result of depletion in genetic variation?
Yes, for example, probably invasive species.
Mm-hmm.
Many situations is less genetically polymorphic population are more evolvable.
And it's not because of multiple introductions, and therefore actually... Mm-hmm.
It's not just episodic interaction, but interactions that can be also purification population of these models of frequency differences.
Yes. Mm-hmm.
But it's just a model. It's just a model.
But topic for the rest of this
Yes, indeed.
So we don't have any crucial questions which should be asked now instead of or somewhere else. So now is the time. If not, so thank you.
Thank you very much. Yeah.